Good evening, I'm Thomas Bosch. I'm speaker of the Collaborative Research Center Origin and Function of Metaorganisms at Kiel University in Germany. Microbes carry out the bulk of biological activities on this earth. Microbes were there first. People have 1.3 times more microorganisms in their body than their own body cells. The microbial community influences how we think and how we digest food and how we defend against pathogens, and they influence our metabolism, our physiology, our behavior. There is a gut-brain axis. They may influence how we behave and even how we take decisions. So it's very clear that this meta-organism research has reached the social, the historical, the philosophical sciences. And since we can transfer microbes from one individual to another, and it transfers certain function and even certain phenotype features, that we are rethinking the term of what is really an individual. All of that, of course, has meanings far outside the natural sciences. And therefore, we thought it's a good idea and it's time to include the thinking of historians, of social scientists, of philosophers in our attempts to understand the impact of this omnipresent microbiome. And so it's my great pleasure and honor to host a roundtable discussion and welcome and introduce my colleagues Hannah Landegger from Los Angeles. She is professor at the Department of Sociology and the Institute of Society and Genetics at University of California, Los Angeles. Thomas Pradeux, he is a philosopher of science at the University of Bordeaux, and Tobias Rees, he is director of the program Transformations of the Human at the Berkgruen Institute in Los Angeles. It's 7 p.m. here in Germany and in France, and it's late morning in Los Angeles. Welcome to all of you to our round of discussion on issues where host microbe researchers in their daily life may not think about, but maybe we come to the conclusion they should think about issues we are discussing now. Before we start our discussion, I would like that maybe you introduce yourself in a 1-minute statement to our audience, and maybe it's a good idea that maybe Hannah starts and then Thomas Pradeux and then Tobias. Hannah, please.
Thank you. It's a great pleasure to be here. My name's Hannah Landegger, and I am a historian and sociologist of science, but I like to say that I speak biology. I did a degree in cell and developmental biology at the University of British Columbia long ago, before my PhD in science and technology studies, and I have maintained the sense that it is important to do both social sciences from in their own terms, but also to inhabit the life sciences and understand scientific concepts as they are unfolding in the present. So my work is both of historical nature, but I also am appointed here in the Division of Life Sciences, and I like to ask biological questions as well.
Thomas.
So I'm Thomas Pradeux. I work at the University of Bordeaux in CNRS, as you said, Thomas, and my work is about is in philosophy of science, and my domain of expertise is immunology. So I work on immunology, the microbiota, and increasingly on cancer. These are the three topics that obsess me. And yeah, I do philosophy in science, which is a way of saying exactly what Hannah just said. I have a background in philosophy and also a background in biology. And I work permanently in a biology lab, an immunology lab, because we think it's important to have philosophers working together with scientists.
Thank you. Tobias.
Hello, thank you very much for involving me in this roundtable. I'm Tobias Rees. I'm Reid Hoffman Professor of Humanities at Parsons and the New School in New York. And I'm not a member of a particular department. In fact, I've worked very hard the last 5 years to not be disciplinary and to not be subsumed by disciplines, because my work is precisely revolving around the inadequacies of currently existing disciplines, given the knowledge that, for example, microbiome research produces. In addition, that's where I spend most of my time. I'm a director at the Berkman Institute of a program called Transformations of the Human that is precisely focused on how fields like microbiome research, but also machine learning or neurotechnologies, are challenging and changing the modern concept of the human as more than nature or as other than machines, and the kinds of complications and the kinds of questions that emerge from that. I'm very much looking forward to discussing with all three of you.
Thank you very much. As a speaker of a collaborative research center funded by the German Science Foundation, Origin and Function of Metaorganisms, I thought I wanted to learn what you think about metaorganism. And the question I would like to ask each one of you is, what does the term and the concept of metaorganism mean to you, for the historian, for the philosopher, for the social scientists? And maybe Hannah starts with, from the historian point of view, what does the concept of the metaorganism mean for you?
Well, I think that it's a very historically particular term, and that so I think of it as a sign of changing times. I think that the history of microbiology is full of all kinds of ways of specifying and taking apart things into species, and thinking about individual cells, and to see the emergence of these concepts of a metaorganism and the ways in which multitudinously different things live together in units. It does seem to me to be a kind of historical shift, and it makes me ask why, and what is it that's pushing us, not just in scientific knowledge, but socially and culturally. What is necessitating this shift to thinking in meta terms?
Interesting. Thomas, how do you see the term and the concept of the metaorganism in a brief statement? I know that you could spend now hours on talking that, but just a brief introduction statement.
So I think this is a very important concept, first. Second, I think it's pretty recent historically, but Hannah will know much better. And I think it's interesting to focus on the prefix a little bit. So superorganism has been there for a long time. I think metaorganism was supposed to refer to metagenomics, at least to some extent, but it's also a very general prefix of meta, as you know, can mean all sorts of things, including just after, like the metaphysics is after the physics. So I think clarifying the meta, I think, would be extremely useful, extremely important. And for me, just to answer your question, I think metaorganism is very important, both from an ontological point of view and epistemological point of view. But by this, I mean the following. It's possible that metaorganisms exist as such. So this would be entities in nature, if you adopt a realist perspective, or they could be epistemological entities, by which we mean that they would not necessarily exist as such, or we would not declare that they exist, but they would be useful entities to think about biological interactions. So for example, we could say, now we cannot do physiology without thinking in terms of metaorganisms, without necessarily claiming that the metaorganism is replacing the organism as the fundamental physiological unit. So I think there's really something to say about what kind of claims or claim we are making when we say we should think in terms of metaorganism.
Thank you very much. Very good. Tobias, what does the concept metaorganism mean to you as a philosopher and an anthropologist?
Yeah, interdisciplinary. For me, the term metaorganism indicates a philosophical problem, and the best definition of what a philosophical problem is, I borrow from Ian Hecking. Hecking said philosophical problems emerge when the space of possibility within which we are used to organizing our thoughts mutates. So the space of possibility mutates and we must think again. And that's pretty much exactly how I understand the term metaorganism. It's the discovery of it's a discovery that led to knowledge, a knowledge that defies how we have been thinking before. So we used to know that there is a concrete delineated individual. We used to know that there is a host, an organism, and that there are microorganisms that are living on the host. And the discovery that on all three levels, microbes, microbial genes are important to the genome, right? We have 20,000 so-called human genes and about 2 million microbial genes that interplay with our genome. On the level of the immune system, where the distinction between self or not self is not quite as simple as it was before. And on the level of the brain, when most neurotransmitters, or at least precursors to neurotransmitters, are made by microbes, that is pretty wild. And the term metaorganism for me is happily imprecise. I like that because it is at this moment in time just an indicator of the need to think again and to revisit and revise several of the concepts that we took for granted as true. This is what an individual is. This is what a self is. This is what an organism is. This is how it's set apart from the environment. No longer work. And I'm curious about how we learn to think differently, what ontological consequences will emerge from that, like what is a self, what is an organism, what is an individual. And I'm curious of participating in this rethinking and in preventing us from stabilizing it too fast.
Thank you very much. So there's the three views which are slightly different, but I think what I hear is that there is a great deal of imprecision and flexibility in using the terms, and Thomas Pradeux would maybe even say this term, this form doesn't really exist, it's just the meaning of something. Hannah sees it as a historical shift. Let's start a little more precise. My molecular cell biology, evolutionary developmental biology work is heavily influenced by the observation from my medical doctors of metabolic disorders and by the fact that we see what I call the last 70 years paradoxon, that despite all our modern lifestyles, we see more and more patients suffering from complex, another very imprecise term, complex diseases. And these metabolic disorders are obviously somehow connected to a connected and a co-metabolism of the microbiome, which has its own life, and the biofilm, and the host, which has its metabolism. And colleagues around us start to think that complex diseases and metabolic disorders may be due to dysbiosis, because the being together with the beneficial microbes is somehow disturbed and causes the problems. So, in other words, I try to understand basic concepts of what's going on in these multi-species associations, but to understand what happened in these last 70 years, the Anthropocene, or a part of the Anthropocene, that this living together was heavily disturbed. Hannah, from a historian perspective, metabolic disorders are something which is typical for our time, or how you would explain the rise of these diseases in the last 70 years?
So, of course, we have historical evidence that symptoms that came to be called diabetes have been around for a very, very long time, or, different distributions of adipose tissues in bodies have existed, of course, for a very long time. But what we've seen over the last decades, really the epidemiology and medical attention has really focused on asking how, why, when, and how can we fix the rise in these metabolic disorders. So I'm not sure that I'm in the business of explaining the cause of obesity as a historian, but I will say that I think that the metaorganism concept actually opens out a way of thinking about the conjuncture of these social, material, historical changes and these biological, medical shifts that we're trying to take account of in the laboratory. So in my talk, I talked about arsenic and the ways that after, more than 70 years, 100 years of industrialization, we see exposures at low doses, chronic low dose exposure to these things as becoming much more prevalent. And that experimental evidence shows that this might be having an effect at the gut wall. Well, as a historian, I could say, well, let's focus in on the gut wall as this kind of historical conjuncture and think about all the different historico-biological forces that are operating at the gut wall. In science, we tend to look for one cause. And we say, well, maybe arsenic is the low-dose exposures to arsenic as a metabolic disruptor is the cause of obesity. But let's look what's residing organismically at that gut wall and its historical trajectories. So you have to include the history of pasteurization. The history of pasteurization has profoundly changed the nature and the number of microorganisms that go into any gut. Let's look at the history of flour milling, the particle size, the fiber component of the fibers that are going into the gut. So at any one moment at this historical conjuncture, you have the historical trajectory of pasteurization, of refrigeration, of flour refining, of cultural attitudes towards digestibility, this sort of push in modern terms that modern food should be highly digestible and easily digestible. And that meets with arsenic at the gut wall and really changes the configuration of who can live there, in what configuration. And so those kinds of interactions, I think, not just impacting the human, but impacting the whole arrangement of the metaorganism that is living in that environment can open out new questions that then may give us some more complex arguments about the cause of something like a rise in obesity-related disorders.
Yeah, I think this is a very important point. The simple-minded, biochemical-oriented microbiologist stumbles on changes, maybe on dysbiosis, on change in microbiome. But the question is what is causing that? And I guess, and I think, and I believe that the historian view is an important one to inform us about possible ways how to think what happened and what is the cause, and maybe even then how can we reverse that. When we remove the microbes from certain model organisms, we get what we call a germ-free signature. And that germ-free signature then is informative of the function of the microbiome. And to our maybe surprise, we find that many of the things which change are metabolic components or the metabolic network. So I'm looking to Thomas Pradeux and asking, when we talk now about not only about the metabolism of the host, but also about the metabolism of the microbiome, and we talk about and we have evidence that all of that really functions together, and by removing microbes, we disturb certain parts of the metabolism of the host, and people have then coined the term community metabolism to indicate that we cannot really talk anymore about a single metabolism which is confined to a given entity which you may call the host. How would you see this development of new terms as community metabolism, codependent metabolism? Is that something real? Or is that just a concept which exists there and may help us in understanding? And in my understanding, it's really something real, because we can return that by what we call conventionalization, and by bringing back microbes in this empty host, we can reverse and we can bring the metabolism, at least to some extent, back in its normal functioning. What do you think about it, Thomas?
Yeah, so I think that the most important transformation in recent studies involving the microbiota is really about physiology and metabolism. So I was a little bit disturbed when I saw that philosophers of science, especially philosophers of biology, when they got interested in the microbiome, they immediately asked questions about units of selection. And I have no problem with that. It's just that I don't think we should start with this question. I mean, I've been working with immunologists, working on the microbiome for years. You, Thomas, you've been working on this question for years as well. I promise the excitement is about the physiology and the metabolism first. And evolution, of course, is important. We have many questions to ask, but my first message would be, let's talk about this physiological metaorganism. I think this is super important. And I think what you both said, Hannah and Thomas, I think is crucial. I think metabolic interactions are absolutely crucial. We are talking here about something where interactions are very intimate, especially at the metabolic level, and we have to understand how it works. And I think this is super important. In practice, people at the hospital where I work, they pay attention now to the microbiome. So of course they have paid attention to the microbiome if they work, of course, on IBD, for example. But people working on autoimmune diseases or inflammatory disorders, they now all pay attention to the microbiome and to metabolic interactions, co-construction, co-metabolism. Even if they don't use the term, they will pay attention to that. And I think this is super important, and this is extremely promising for the future. I, again, will be a little bit careful in the vocabulary, but it doesn't mean that it is not important and interesting. Quite the contrary, I think this is super interesting and super important to really understand what's going on causally. But for example, this idea that dysbiosis would be a good explanation for what is going on, I'm not so sure about that. I mean, it might be the case that dysbiosis is going to do an interesting explanation for what's going on. But my message would be, even if there is nothing like a universal or even common dysbiotic state to several people, because dysbiosis is very complex to define for several reasons, especially because one question is are we talking about my dysbiosis or the dysbiosis of my group or the dysbiosis of every human being, etc., etc. What I would suggest is that even if we have difficulties defining dysbiosis, it will remain super important to pay attention to metabolic interactions when we want to understand a specific disorder in a specific individual. So what I'm saying here is that this co-metabolism thing is probably one of the main topics that is going to be explored, I think, in the future in this big domain of microbiome studies. I think this is absolutely clear.
So I'd like to say that thinking about our terms and our structures of thought, as Thomas Pradue points us to do, is very, very important, because as we shift into a realization that there are all kinds of entities that were not noticed before or were not given significance, and it's changing the way that we think about causality, it's very important to practice reflexivity about those concepts and where they've come from. And so one of the things that I've seen in looking historically, for example, is in the rise of concepts in the 20th century around genetics and metabolism, around executive functions and housekeeping functions, even about the separation of the body into systems like the immune system, the endocrine system. They become supernaturalized. We come to think that they transparently represent the world. But sometimes that really gets in the way. And so now in immunology, for example, you see the emergence of immunometabolism and realizing that some intermediaries of the Krebs cycle, for example, that were simply seen as inert intermediaries are determinants of immune cell differentiation, for example, and being really reflexive that it's not that was difficult to see before technically in some way, but that it did not fit a frame of thought in which some entities had all the agency and some entities were the ones that were acted upon. When we can see that framework really shaped the experimental questions that we were able to ask, we can be a little more reflexive and ask in the present, okay, maybe we've shifted that framework, but what are we giving the leading role to? Who's the protagonist? What's the thing that is just passive and inert in this picture? What are we leaving out? What are we maybe not asking that we should? So I think this kind of historical and philosophical reflexivity about these terms that organize the experiments that we do can be really, really useful in asking unexpected questions.
I fully agree, and I would like to turn a little bit to more philosophical question, and by using your term, which both of you, Hannah and Thomas, have used several times now, and you call it entity and different entities. And I turn to Tobias now and ask, can you really identify and then separate the different entities which are maybe making the phenotype, a specific phenotype? Can we separate that? Can we separate the human from the omnipresent microbiome in terms of functionality?
Well, right now I don't think so, but I would like to frame the question maybe in a much more specific context. So we talked about disease, we talked about dysbiosis. I want to use the term pathology for a second or the term pathogen for a second, that will allow to make that more specific. For a long time, there was a very clear understanding that there is an organism, there is a pathogen that entered that organism. The pathogen was the disease. It was what had to be sort of identified and had to be gotten rid of, so to speak, in terms of drugs or surgery. And the microbiome troubles that significantly, right? It does something to the concept of pathology, what pathology is. And it's directly related to your question, Thomas, because there were two entities, an organism and a pathogen. And the question is, are there still two entities when we talk about pathology today? And when you take strep B, we all have strep B in our throat right now, and we know that we don't get sick by them, and there is some speculation, at least, that they actually may fulfill essential functions for our throat well-being, so to speak. We had a long discussion at one of the CFAR workshops whether TB would today be a commensal or a pathogen, so to speak. And it was a wild discussion because, of course, the pathologists all insisted it's a pathogen. And then there were some who said, no, it's actually not. So the reason why I mentioned this is that with the microbiome, it appears with dysbiosis in particular, that what we used to call pathogens might be actually a normal part of the microbiota, of the metaorganism, if you will. But when a dysbiosis happens, i.e. when one microbial population overgrows the others, then you have disease. But that means that there is no more clearly identifiable entity. That means that disease is, so to speak, a relational configuration, and that one has a new pathogen concept, so to speak. The reason why I find that interesting is that Hannah said that many of the concepts, like immune system, for example, get naturalized very quickly. And we tend to think that the individual exists. It's like a freestanding, clear thing. But these are historical categories. They're not ontological categories. And so the power of the microbiome is that it generates knowledge, in my understanding, that undermines some of those ontological categories. And the strongest level at which this occurs, I think, is the human. We truly tend to take it for granted that we're sort of more than nature, that we're kind of set apart. The whole idea of the human sciences or faculty of arts or the social sciences is that human reality is set apart from biological reality, which is why it needs its own kind of science, as if there were this split. But from a microbiome perspective, the idea of the human as such, as set apart, is sort of a sterile idea that only exists in the petri dishes of the human sciences, if you will. And I find that provocation incredibly exciting, not because I'm against the human sciences or the faculty of arts, on the contrary, because it gives microbiome research a philosophical quality or dignity that it did not necessarily have before. And thinking about this together is, for me, extremely exciting.
Thank you very much. I'm sure that Thomas Pradeux has a different view on that. But before giving Thomas the reply to your thoughts, Tobias, isn't it so that because the microbiome is not visible and it became visible, and that changed so much of our thinking of how organs function and how diseases, how we should consider disease, and we talk about patients as metaorganisms, etc. Isn't it because the microbiome is just so different, or so small cells and not visible, but our body actually is home to many other organisms, and we never would have come to that conclusion when we think of the worms we have in our body, and they may do no harm at all, but they are just with us for years and years and years. And so what makes is there then a good reason to just concentrate on the microbiome and the host, or must we think that in a more broader sense, or just say that, these are real different entities and they are just together for good reason, and also evolution has selected for that. But I feel a little that the microbiome these days gets a very special meaning and puts the microbiome apart of any other eukaryotic colonizer which is in our gut or which sits in our skins for a long time. Tobias.
This is addressed to me, yes. So let me first say that first I agree. Yes, there is and I think you say at least two things. One is the microbiome is just a new discovery, and so there is new knowledge, and that is sort of, in my language, an opening because it contradicts prior conceptualizations of things. And then the second point that I hear is, are we not getting too much stuck on microbes, qua microbes? There are many other things. Community metabolism, actually, for that matter, is another thing. It's not specific to microbes. I think the concept was mostly used in aquifers or aquatic systems, and clearly that's not only focused on microbes. It's like a fully integrated system that could be looked at as close. There are animals, microbes, but also plants are kind of looked at as a single metabolic system. So from my perspective, I look at the microbiome research field as a conceptual field, right? So I try to basically look at it in the sense of what kind of knowledge emanates from it, whether and how does this contradict prior ways of thinking, whether these are prior ways of thinking that are implicit in biology, the concept of the individual, for example, or whether these are concepts that are sort of broader, like in terms of what's the human, what's nature, what's technology, what kind of alternative ways of thinking conceptually, experimentally do emerge from microbiome. But my more principal search, if you will, is for a single descriptive language, not as an ontological feature, but as a descriptive language. Hopefully there are many of these descriptive languages that allows us to think about humans, technology, and nature as fully integrated, so to speak, as not set apart. What would that look like? What has the microbiome to offer? What does community metabolism have to offer? What would biogeochemistry have to offer? And that's the perspective on which I'm thinking. And maybe just one quick wrap, because I'm really curious about what Thomas is thinking about. That is, so historically, I guess one of the first places where the concept of the individual emerges is in Leibniz. And there is this famous story how he walked with the Contessa of Herrenhausen in Herrenhauser Gardens in Hanover. Through the garden, and she says, no two leaves have the same structure, and he says, that's total rubbish. And they begin picking leaves, and they actually pick thousands of leaves, and some of them that they back then picked more than 300 years ago are still preserved, and they studied them, and of course no two structures were the same, and Leibniz was completely confused, because how can a single substance give rise to all of these different forms? And so that's when he introduces the idea of the individu. What makes each leaf special is that in which it differs from every other leaf. And I think on the level of the history of thinking, that's hugely interesting. First, because it's the first time that a concept of the individual is made available to humans so that they can begin experience themselves as individuals. And the second reason why it's so interesting is it's the leaf that is the model for the human, so to speak. It's leaves. And the concept of, in biology, when you take Wolf, for example, who invented the concept of epigenesis, is also with leaves, right? So I find that wild. So for me, the individual is simply a historical category of thought. It has no ontological truth content, zero. And so now I go to the microbiome, and I wondered, from a microbiome perspective, do individuals exist? Is that even a meaningful category? Should we assume that individuals exist and then look at the microbiome? Or should we just look from the perspective of the kind of indistinguishability between the host and the microbiome that then emerges? So this correlation between thinking or categories of thought and being, and what being can be for humans, is something I'm very curious about. And I wonder what the two of you actually make of this in your work.
Thomas, that's up to you now. The indistinguishability, I guess you will disagree. And the other thing which I would like to raise is what Tobias just said. If leaves are a model for humans, and in a philosophical sense, microbes are also used as model in many ways now for human behavior or thinking, even for human mind. Is that something which makes really sense, and is that something one should encourage to go on, and is it informative at all for our way of trying to understand better the human? Maybe first Thomas.
Well, there are many questions here, all of them very, very important. I tend to be extremely pragmatic. I think that everything that is fruitful should be used and tried. So, for example, if we think that we can say something very interesting about behavior by using a model which is an unusual model, I think we should try and see what comes out of it. So I'm very open-minded from this point of view. In terms of individuality and all the rest. So the first thing, of course, is that our mental categories are absolutely essential in the sense of important for the kind of ontology we are going to develop. So that's absolutely obvious and absolutely essential. Why is that obvious? It's obvious for the humanities. I mean, we've known that forever, but biologists and scientists more generally, very often, they don't think about that. It's not that they don't know it. It's just that they tend to think that the categories are real and have been there forever. So doing a historical and conceptual analysis is extremely important, just to remind them where the idea comes from, where the concept comes from, where the term comes from. I think that is extremely important.
Of course, Hannah, you would of course very much embrace that and support that point of view. Yeah.
Well, I'm going to poke a little bit of fun and say that perhaps Leibniz should not just have examined the leaf and looked at the leaf and compared one leaf to the other, but maybe should have tried eating the leaves. Do you come from do you get to a very different spot if you say the leaf is nothing but its relation to the organism that I am? And so you asked this question, right, about entities and separating entities. And what if what a leaf is only its relationship in a processual way, its relationship with the insects and the microbes that live on it, and when ingested, what happens to all of the parties in that relationship. So maybe philosophy would be different if it had considered everything by ingestion rather than visual consideration.
Can I say just something on that? I'm completely intrigued by this. This is going to be even more playful now, or the same degree. But I just follow Hannah's invitation. We all know about the importance of René Descartes and je pense donc je suis, I think therefore I am. But at the microbiome, and at this, the idea that thinking is basically a neurochemical act, that the neurotransmitters you have depends on the bacterial populations you have, which in turn depends on what food you eat, right? Because food's the only way to sort of control which bacterial populations you have. But it also depends on the way the food was produced, if there's lots of synthetic nitrate, no flavonoids, etc. So you could take the sentence, I think therefore I am, and say, how many was Descartes, so to speak, at the time he was saying this? What did he eat, and how did the food that he ate lead him to say what he said? So you can break down the modernist concept of the human as, cogitans versus res extensa, and completely undermine it. You could write a brilliantly you could write a brilliant provocation around this. And that would, for me, beautifully capture, like with Leibniz and the leaf, and try to eat it, the power of the microbiome and its implications for philosophy.
Thank you very much. We are now in a very in a wonderful area of philosophical thinkings. I would like, also in the interest of time, I'd like to come back a little bit more concrete, but also entering still different territory. And I make a very strong statement, but dysbiosis, or whatever we agree, we think of that, which has consequences for health and disease, is a problem of your socioeconomic status. And as we know, many of these complex diseases, it's a societal problem, and also, by the way, the current pandemic, and that means understanding and trying to get the mechanisms behind has also a strong ethical issue, and at least for me. But again, this is not really considered in our daily host microbiome research. Would you agree that one should consider and maybe emphasize or do more research on the socioeconomic consequences on the development of these disorders, metabolic disorders? Hannah, what do you think?
Yes, I think the current pandemic and these intersections between a novel infectious disease and long-standing and long-building questions of chronic diseases has really done a bit of a number on the way that we foreground and background things. So the so-called underlying condition has been very much foregrounded here as we see the patterning of the disparities in outcomes from COVID-19 by social, economic, racial, cultural parameters. And I think that this really, and it's not that these things were not known before, and we knew already that outcomes from influenza are shifted by diabetes, for example. This is not completely new, but I do think that it lends some renewed urgency. Perhaps the idea of a chronic disease that is long-lasting and persistent and always there has drained it of some of the urgency that it perhaps should have, and that the current situation is really highlighting that this is a question for medicine, but it is also a question for politics and ethics. How should we live? What is the basis of human well-being? How is it that we can ensure that humans can flourish in their nutritive and stress environments? And then the reverse of that question is, what is it about the configuration and the intersections in any one body that social arrangements are so manifestly embodied? And so I don't have answers to those questions. I can see the outline perhaps of saying we need to develop a kind of metabolic ethics, if you will, ethical questions about how we should live, or political questions of how we should regulate, where the ability to flourish, whatever that means, with a working biosis, a functioning biosis, is at the center of directing us in terms of what decisions should be made. There are many, many substances in our world which go unregulated and uncounted, perhaps because they don't cause genetic damage. So we think, oh well, they're not important. They're not injuring the most important central stuff, right? Well, maybe we need to really bring into view some of these things that are metabolic disruptors, and say we need to shift our traditional centers of concern to pay more attention to this, both at the scientific level but also at the social level.
Metabolic ethics, Thomas, what does the immunologist say to that? I mean, in terms of inequality and dysbiosis and these entities which need a certain environment, and this environment is not equal for everybody. What do you think?
Yeah, I mean, I definitely agree with what Hannah just said. I think this is extremely important to pay more attention to all these interactions, environmental exposures. I think in terms of socioeconomic situation, inequalities, there's a lot to do about this. So for example, if you consider endocrine disruptors or metabolic disruptors, if you want to investigate that term, I think we will find many inequalities in terms of how you're exposed to that kind of environmental influences. I think the impact of that on our health on our microbiome as well will be extremely important. And it's very important as well to say that the economic interest there, the industrial interest there are absolutely huge. So it's also extremely important that we researchers, insist that, for example, take plastic. I mean, this is a huge thing, not just in terms of pollution, but I think it's also a huge thing in terms of what people are exposed to. And I'm pretty sure that people have investigated that already and will investigate that even more in the future. And I'm pretty sure that the degree of exposure of people will be very much dependent on where they are in the society in terms of socioeconomic status. And I think it's very important that we researchers in general, philosophers, scientists, medical doctors, anthropologists, historians, I think it's very important that we constantly also fight on where exactly the money is, where the money is invested so as to sometimes create a pseudoscience that says a lot of things which are used and reused by some industries and some medias. And I think one of our roles is to do the science that is the real science that will accumulate enough studies to say, okay, this is very clear, there is this effect, or there are these effects, and there is a difference in the way different people in terms of socioeconomic status are exposed to this risks of danger. I think this is part of our job. I cannot do that myself because I'm not an epidemiologist or a medical doctor, but I can help to maybe frame the question in such a way that for society, it becomes clear that we are not going to just let a few studies, 5 or 6, be described as the demonstration that something, for example, doesn't hurt, or that kind of thing. So I think this is very important that we also investigate that kind of questions.
Thank you very much, Thomas, for this very clear statement. Tobias, you want to add something here?
A little while ago, I had the honor to write an essay together with a synthetic biologist from Stanford called Drew Andy, where we discuss, should synthetic biology go ahead and build synthetic cells? Should there be a synthetic human genome where you write code on the computer basically and then print the DNA and put it in a cell and you have a human? And we decided that we're hugely excited about the possibilities that this brings, but that we're simultaneously very worried because engineering has happened almost exclusively from a human-centric perspective, so to speak. So could one engineer from a whole Earth perspective? What would that look like? And the reason why I bring this up is the question of the social and how to frame it, whether what's at stake is society or whether what is at stake is, so to speak, community metabolism. What's like the unit of interaction or the unit of intervention, that we have. So for example, when you look at the history of social intervention, the idea was literally that one intervenes in a social fabric. It was sort of society that was at stake, a human playing field. It was not about microbes. It was not about organisms, so to speak. What we today call social is actually often biological, right? And so as I'm interested in how the concept of the microbiome opens up possibilities of thinking about humans or about justice or about flourishing, about in public health, if you will, in new terms, like in new ways of framing that. I wonder if society is actually, is so to speak, the right term, because the unit of intervention would have to possibly be an environmental unit. It would actually possibly have to be an industrial plant that is close by. It would have possibly to be food. Or agricultural systems. We all talked, or Hannah, Thomas and I talked to Kate Brown a little while ago, and the idea of the community garden and why this could be so extremely important right now. And when you frame it like this, then the question is whether society is the unit of intervention or whether a garden, so to speak, is the unit of intervention. It's very different. And this is a shift in thinking that I find provocative, to be sure, but if well-being is at stake and the human cannot be separated from the flora, then we have to wonder what the right unit of intervention and problematization actually is.
Thank you very much, Tobias. This is a wonderful conversation which could go on for quite a while, and we would enjoy it very much. Here is a historian, a philosopher, and an anthropologist, and the time is comes to an end now. But before we close that wonderful discussion, I would like to ask each one of you. We are certainly, Tobias says, we are anti-disciplinary oriented, and we are certainly particularly prone to talk to people from different areas. What would you, Hannah and Thomas and Tobias, tell a junior researcher when you want to encourage him or her in listening to different disciplines? Why should that be for the junior investigator efficient and informative and rewarding? What would you say if somebody approaches you and you want to encourage her or him listen to different areas and it will enrich your thinking? Hannah, what you would say is, make it short, a few sentences, and then Thomas and then Tobias. Hannah.
So I would give a couple of pieces of advice. One is to go to talks, even if you don't think that they're relevant to your work. The second is to read widely in different genres, to explore perhaps what literature and poetry, or even something like statistics or epidemiology if you're a molecular biologist, to explicitly cultivate the crossing of scales. Because if you look at the history of science, many of the moments of opening and rupture come from someone saying, I do not accept the assumptions that are on the table. I see it differently. So how can you break the frame of perhaps your seniors who are saying this is the way everything is organized? How is it that you see assumptions that are by their nature embedded and invisible? Well, you have to think across and around and cultivate this kind of reflexivity around the question of how do we know what we know? Why are we asking this question? What in this scene is given all the credit? What is being disappeared from this scene?
Thank you very much. Thomas, what would you say to the junior investigator?
Well, that's just perfect because Hannah said it all. So I would say exactly the same kind of things, and I would very much insist on examples from history of science. So in my case, this is not that I have a huge knowledge, but in the fields I know, we can demonstrate pretty easily that big changes happen when people disagree with underlying statements or underlying assumptions of the community, exactly as Hannah said. And in almost all cases, these people who criticize underlying assumptions are outsiders. Of course, they are scientists to some extent. They've been trained in science, but they're different. They did two things at the same time. They are microbiologists, they arrive in immunology, the other way around. Or they start talking about the microbiome, even though they are developmental biologists in the first place. So I think this is exactly what I would use as a series of examples where you see this is extremely productive if your aim is to change science, not just to do science. And of course, here as well, there's no boundary between the two. But if your aim is to do something very novel in science, the kind of interdisciplinarity we're talking about is super important. And we're not just talking here about philosophy and history of science. I mean, I encourage immunology students who work with me to do ecology, for example, or evolution, or physics for that matter, even though I'm clearly not an expert in this domain. So I think that kind of, open-minded approach, I think is super important and super productive. But then the question you did not ask, Thomas, and that will be for another time, is how we convince departments to really pay attention to that kind of people. And I think what Tobias said before is extremely important. I think we're in a situation where it's very good to encourage the young to do that. But we, the more senior people, we must rethink very carefully how we encourage careers which cannot be summed up by saying, oh, okay, now I know you're a historian, or now I know you're a biologist. For all these people who have these very rich backgrounds, we need to find I don't know, bridges between departments, or maybe departments of nothing, or departments of interdisciplinarity, I don't know, something like that, that will allow them to thrive with the kind of backgrounds they have.
Thank you very much, Thomas. Tobias.
Yeah, it's indeed very easy to follow up on both because it's all said, kind of. I think that all four of us, in that sense, are accidents of a system that is set up to not produce the accidents that we are, basically, right? And for me, when I began to recognize that this modern notion of the human, that we're more than nature and that we're irreducible to mere machines or mechanism, is reflected in how the university is organized. I was shocked, basically, because the Faculty of Arts, that is about the human insofar as it cannot be explained in terms of the natural sciences or engineering. It's a Faculty of Science, which is about nature, and then there's a Faculty of Engineering, which is about machines. But as we live in a moment where we learn that we're not more than nature, we are microbial, so to speak, whatever that exactly means. And when we live in a moment where machine learning and quantum computing, at the very least, put in suspense the question, A, what is a machine? And B, are humans more than machines, or what would that actually mean? We need to have people who can traverse these three fields, and we must create new universities or reform the university from within, or have a full integration of departments where young people can be simultaneously a philosopher, a natural scientist, and an engineer, so to speak. That must be possible. If we don't do that, we continue to educate in 19th century terms, and it would be just a sad story.
Thank you very much. That's a wonderful ending of a really exciting discussion. An evening hour here in Germany and in France, and a late morning hour in Los Angeles. I think the challenge is very clear, but also the prospect where this could lead if we succeed in reforming the structures, in teaching the young generation. I stay in my discipline for the very last moment in developmental biology, and it's very clear for me that in developmental biology, new things happen when unexpected layers come together, and the developmental biologist calls that an induction. We have to induce our systems that they are able to cross boundaries outside boxes, outside silos. I would like to thank Hannah Landecker, Thomas Pradeux, and Tobias Rees for a very inspiring discussion on a topic which started once with understanding the interaction between microbes and host, typical biochemical molecular problem, but which has so many different aspects. And to understand that, and in the end to understand human, we need this interdisciplinary thinking. Thank you so much. Thank you for joining us. Thank you for the time which you have now spent with us. And I wish you a very good afternoon or a very good night. Thank you very much. Goodbye.
Thank you.
Thank you very much.